So this right here, we're going to get to the video in just a minute. This picture that you see right here is something that you're going to see later in this video if you stick with it. There's something I do want to clear up about this video. It has something to do with the way that I teach this particular topic to help people understand it. We're going to talk about something that's basically referred to as a depolarization. Okay, this right here, this right here is called a depolarization. And what depolarization is it's when you take these positive charges, in this case it's going to be sodium, and you move them from the extracellular fluid into the cytosol. That's what a depolarization is in this case. And I found over the years that if you get somebody to understand that, yes, sodium moving down the muscle cell membrane, that is the propagator of the action potential. If you can get somebody to at least understand that, that's the best thing in the world. Okay? There are some other details that we can add in a little bit later that I don't feel we need to add in at this point. Okay? So if this next thing confuses you, don't worry about it. We'll get it much later. Okay? Yes, the sodium is going to propagate this action potential. Okay? But there also are some potassium ions. So for example, if I'm looking at this, let's say I look at a particular point. Let's look at this point right here. Let's say I look at this point right there, okay? And let's say I'm looking at a coordinate diagram. Let's say this is the time axis. Well, what's essentially going to happen is at some point in time in that region, sodium is going to move across, okay? Sodium is going to move across, etc. Well, I usually don't teach this because it just confuses people. But sometime t, a little bit later in this region, notice I'm moving across because it's at a later time. Potassium is going to move across as well, but in the opposite direction. And you can hopefully see why I normally don't teach that at the same time. It's because it's a little bit confusing to students. So what I want you guys to understand is that sodium is the propagator of the action potential. Okay, sodium propagates the action potential. Okay, that's what we're going to focus on in this video, and that's the reason I've taught it this way. Later on, what we're going to do when we actually talk about action potentials in more detail, we'll mention the potassium, that there is some potassium flux across. But the thing I want you to get out of this video, I want you to understand how sodium influx propagates the action potential. That is the biggest key from this video. Okay, you'll start to see the method as we go throughout this video, so focus on that. Understand that when we do get to this figure and we start to go through it, that there are going to be some potassiums that move across, but that's not the big picture. If anything, potassium helps to terminate the response. It terminates the response in that region. We don't need to concern ourselves with that right now. Okay, so if you're watching this and you don't go to this school, understand that whenever I move sodium across, in general, at some time t later, okay, there's going to be a potassium that moves across. Okay, that's not what I feel is important. I feel we have to focus on the sodium, because if you understand that sodium propagates the action potential, you're golden. So I just focus on that, but we'll talk more about potassium in later weeks when we do the action potential. So I just wanted to clear that up before we get into the meat and potatoes of this video. Welcome back, guys. So you just had your exam, and so at this point the exams are graded, the grades are posted on Blackboard. At this point we're going to start a new topic, and in general we're going to be talking about muscle physiology. We're going to be talking about the events that cause muscle contraction, starting with the motor neuron. We'll talk more about that. And in general for today, what we're going to be talking about are the events that happen at something called the neuromuscular junction. Okay? Well, what is the neuromuscular junction? Well, before we get any further, what we essentially have is this picture right here. So this structure right here, okay, this is called a motor neuron. It's a component of the motor neuron. So it's a component of the motor neuron. Specifically, it's the axon of the motor neuron. Down here, this is essentially part of the muscle, okay, the muscle cell, and this specific part of it is referred to as the motor end plate. And so if I had to define for you what the neuromuscular junction is it's a physical junction, and in fact it's this space that's kind of in here, right? It's the junction or the synapse between this axon of the motor neuron and the motor end plate of the muscle. So if you take the space or the synaptic cleft between those two structures, that's what the neuromuscular junction is, and we're going to be talking about the function of what's going on here. And it's quite a difficult topic when you first hear it, so hopefully we'll break it down into some manageable steps. Okay, so in these first two slides right here, I'm going to go over the very general physiology of what's happening, and then we're going to break it down and hopefully understand it a little bit better, because this can be at first a pretty frightening figure. Okay, so one of the challenges is picking a place to start. And in A&P here at this school, we don't start with the action potential. We just kind of say, oh, well, an action potential, is going down the axon, and we'll leave the action potential for a different week. And the action potential is going to be traveling along here, right? And it's going to hit something that I want you to pay close attention to. We're going to be talking about it. This structure that I'm talking about is called a voltage-gated calcium channel. Okay, these types of calcium channels have some very important implications in muscle contraction. So at this point, I don't really expect you to know what an action potential is. That's going to be the topic for 2 weeks from now. Okay, but suffice to say, we usually kind of draw it as this little lightning bolt that's coming down the axon, and it's carrying some charge with it. And so what it does is it comes down here through the axon and it activates this channel right here, which is referred to as a voltage-gated calcium channel. Now, before we go any further, all this stuff out here, okay, this is extracellular fluid. Okay, so we have several ions that are going to be important in the extracellular fluid as we progress through muscle contraction. Number one, probably arguably the most important one when we start getting into the action potential, is sodium. So it turns out that there's quite a bit of sodium here in the extracellular fluid. There is also a considerable amount of calcium, Ca2+. Okay, one of the key things here is that whenever the action potential activates this voltage-gated calcium channel, as you see right here, calcium is going to diffuse from the extracellular fluid into this region of the axon. This is essentially the cytosol of the axon. So this is sort of, we took off the extracellular layer of the axon, and now we're peering inside of it to see what's going on. Okay? Through a mechanism that we're actually going to go into, when calcium influxes from the extracellular fluid into the cytosol of the axon, you're going to get exocytosis of acetylcholine. Okay, so these little dots right here, okay, these are a molecule referred to as acetylcholine. That's a neurotransmitter, and it's the neurotransmitter that activates muscle contraction. So if you ever want to have muscle contraction, you have to synthesize acetylcholine, you have to package it into these vesicles right here, these are vesicles, and then you have to exocytose them through mechanisms that you talked about when we did exocytosis in both lecture and lab. Okay, so what's the key? Whenever the action potential along the axon activates the voltage-gated calcium channels, calcium influxes from the extracellular fluid into the cytosol of the axon, and that triggers exocytosis of the acetylcholine. And then from there, what's going to happen is, in fact, let me keep that on the same slide where all the markings are for now. You have these structures right here, which are basically called acetylcholine receptors. Okay, and the idea with the acetylcholine receptors is these acetylcholine molecules, all of these guys right here, these acetylcholine molecules, they're going to come over here, and we'll look at this in more detail, and they're going to bind to the acetylcholine receptor. Okay, and when they bind to the acetylcholine receptor, keep in mind there's some sodium in here, right? There's some sodium. And when they bind to the acetylcholine receptor, essentially the sodium is going to move through here into the motor end plate cytosol. And that's going to trigger some events, and we'll see how it works in a few minutes. But hopefully that gave you a broad brushstroke of what's essentially happening here at the neuromuscular junction. Now let's sort of delve into the details. We have this action potential moving along the motor neuron. Well, essentially, and it's obviously more detailed than this, essentially what the action potential is it's consecutive sodium ions moving into the axon. Okay? It's sodium ions moving through. Okay? I want to make sure you understand that very well. The action potential, what it literally is it's sodium ions undergoing diffusion from the extracellular fluid out here into the axon. So what's exactly happening in this picture with the voltage-gated calcium channel, which is what we essentially talked about a little bit in the last slide. So what you have to sort of imagine is this right here, okay? This is sort of the axon. I'm going to try to kind of sketch the axon end bulb. It doesn't look exactly like this, but this is sort of right here. This is a representation of the axon. Okay, so this right here, this is the end bulb. Okay, so this is all axon. Okay, and then out here, this is extracellular, the synapse. These synapses out there, right? So here's the basic idea of what's happening here. Okay, basically the action potential is going on. The action potential is the opening of those sodium channels, right, that I mentioned over here. Remember that the action potential results from opening of sodium channels and sodium moving from the extracellular fluid into the axon, right? And you have these proteins that facilitate that. So there's lots of sodium out here. And then the sodium, through the action potential, through a mechanism we'll elucidate later, moves here into the axoplasm, right? This is intracellular. This is the cytosol of the axon, or the axoplasm, right? And then what's going to happen is the action potential is going to keep moving along here, right? Sodium is going to diffuse from the extracellular side into the intracellular side, right? So sodium comes through here, right? And so forth. And I think you may have an intuition that what's going to essentially happen is you're going to get a lot of sodium built up kind of around this area, perhaps, right? You're going to get a lot of sodium built up. Now, essentially what's happening here is, what are you building up? You're building up a lot of positive charge, because sodium has a positive charge. So sodium starts accumulating around here. You've got a lot of positive charge now here in the cytosol. Now, here's where it gets really important, okay? There is a subunit on this channel right here. It's referred to as subunit 4. Very important subunit here when we're talking about the voltage-gated calcium channel. This subunit is called a voltage sensor. What is a voltage? Well, a voltage essentially is when you have a difference in charge between 2 points in space. And so what ends up happening is, at rest, remember there's no sodium out here, but as soon as the action potential occurs, you start building up all this sodium. Well, it turns out that this subunit right here, this voltage sensor, it can sense positive charge. Okay? This subunit can sense positive charge. Okay? In other words, it can sense all of these little sodiums out here, right? And as soon as the voltage sensor starts sensing the buildup of sodium, the buildup of positive charge, this protein, this channel changes conformation. Okay? That's what we're essentially going to look at in this slide over here. Okay? So here's kind of the setup for the voltage-gated calcium channel. Okay? These same channels that are over here. Okay? Before anything happens, okay, you have this vesicle. Right, just the average vesicle that you've talked about in maybe previous classes or even previously in this class. And in this vesicle, at the neuromuscular junction, there is a neurotransmitter called acetylcholine. We typically abbreviate it ACH. So there's a lot of acetylcholine in this vesicle. Okay, it was previously packaged into there. Okay, this is the voltage-gated calcium channel. This is as well. This is the voltage-gated calcium channel. Okay, and you see here all these little proteins here. You don't really have to know all that stuff. Okay, you don't need that to have a general understanding. What I want you to understand is, notice there's a protein right here. Okay, this protein is called synaptobrevin. Okay, notice that along with synaptotagmin, look what's happening. This vesicle through these proteins, notice it's attached to the channel, right? The voltage-gated calcium channel is attached to the vesicle. And this series of proteins like GK and SH3 and C2A, C2B, this is what we call the tether. Okay? This is called the tether, or it's referred to as a tether complex. Okay? So what this means is that at rest, before anything happens, the voltage-gated calcium channel, okay, is attached to the vesicle. Okay? And also just keep in mind that this, in this case, is the extracellular fluid. This is the axoplasm, or the cytosol inside the axon. Okay? So now the question is, what happens when calcium influxes, right? I just mentioned that when the voltage sensor senses the sodium from the action potential, the channel opens. And so what's going to happen is, you guessed it, calcium, Ca2+, is going to move through this channel and go into the axon through facilitated diffusion, essentially. So when this channel opens, calcium moves through. Now we're essentially going to see what happens when that occurs. Okay, so here's the voltage-gated calcium channel. Okay, in fact, in general, there are two of them, but you don't really need to worry about that. So calcium essentially is going to move through here into here. Okay, now if you go back to the previous slide, notice I had this protein right here, which technically is pretty important, but I'm not going to have you really worry about it so much. It's called SNAP25. Okay, it turns out that these proteins right here, synaptobrevin, but specifically synaptotagmin, okay, it has a really high affinity for this SNAP25, meaning that given the opportunity, synaptotagmin would like to bind to SNAP25. Well, it turns out that when calcium rushes in here, calcium facilitates the binding of this tether onto SNAP25. Okay, so this protein right here, this complex is SNAP25. So when calcium rushes in here, the vesicle is attached to the synaptobrevin and the synaptotagmin and all that stuff, right? All this business over here is now brought to SNAP25. And so you can imagine what's going to happen if there's two regions that do this. The vesicle essentially is going to be pulled in this direction. Okay? It's already attached to the channel through the tether, right? But if calcium comes through, there's a change in conformation that sort of forces all of those proteins to be drawn towards SNAP25. And that's going to pull the vesicle towards the membrane. Okay? And just keep in mind, there is lots of acetylcholine in here. Okay? Like I mentioned, acetylcholine is in here, but it will diffuse out. Now, what happens is through a spontaneous process, once these tether proteins are brought towards SNAP25, and the vesicle basically makes contact with the membrane, it fuses. Okay? And it has this little pore right here that will eventually get larger. But the whole point here is that this acetylcholine will exit into the synapse. Okay? This acetylcholine will leave. This one will leave. All of the acetylcholine that are in that vesicle, they will leave. Okay? The question is, what happens to the acetylcholine once it gets in the synapse? Now we're going to find out what that is. So I'm just kind of showing you the general situation of what's happening. Now we've already seen the vesicles undergo exocytosis, so this process was exocytosis, right? So now we have this situation where I have all this acetylcholine here, these little red dots, in the synapse. Okay? What happens to them? Well, I can answer that, and I can answer that using these little proteins that are right here. These proteins that are in the membrane, which is the motor end plate, these are called acetylcholine receptors. Acetylcholine receptors. And acetylcholine, when it diffuses across the neuromuscular junction, the synapse, it will bind to these acetylcholine receptors. Let's look at that. So up here, up here, this is basically where the axon was. Axon was up there, right? And then I have exocytosis, right? I have exocytosis, acetylcholine gets released. It diffuses across the synapse, right? And it's going to end up binding to an acetylcholine receptor. Okay? This little green dot here is representative of acetylcholine. If you wanted to see it on a molecular level, this is acetylcholine as well. Okay? So here's kind of the gist of what happens. Okay? Whenever acetylcholine binds to the receptor, okay, in this case it's a nicotinic acetylcholine receptor, okay, when it binds, the channel opens, okay, and this guy right here, sodium, moves through. In general, it moves this direction. In other words, this right here, this is the muscle, right? This is the muscle. Go back to this picture, right? We're at this point talking about the stuff that occurs here, right? In the motor end plate. So out here's the synapse. This is extracellular fluid, right? Sodium's high out there, so sodium will diffuse through here into the muscle cytoplasm as soon as acetylcholine binds to the receptor and the channel opens. Okay? So hopefully that makes a little bit of sense. So acetylcholine binds to its receptor, the channel opens, and sodium moves through. This type of situation, by the way, where some kind of molecule binds to a receptor and opens a channel, okay, and particularly when the thing that moves through is an ion, this is called, in fact, this channel is an example of a ligand-gated ion channel. Why is it called that? Well, acetylcholine here is considered the ligand. Okay? A ligand is just really, in biochemistry or physiology, it's any molecule that binds to a receptor. Okay? So it's ligand-gated because it only opens, the channel only opens when the ligand's bound, right? When acetylcholine's bound, and it's an ion channel because sodium moves through, right? This is a sodium channel. Okay? So when the ligand, acetylcholine, binds to the receptor, sodium moves through. That's going to be really important when we start getting into the next few processes that are going to occur along the muscle cell membrane. So we're going to kind of see a process that kind of goes in this direction across the muscle cell membrane. Now, this type of ion flux across the membrane, it's ligand-gated. Okay? But what you can sort of imagine happening, just like we've seen before, is assuming lots of sodium is moving through, I'm going to start building up lots of sodium here. This is going to be sort of a common motif that we're going to see. So the sodium buildup is going to go in this direction. So sodium is going to be building up. Okay? That's going to lead us to this. Okay? So what you have to imagine here is this right here. This is the muscle cell membrane. Okay, the muscle cell membrane. And this is technically is a channel that kind of, when it's folded up nicely, it looks like this, but they've sort of just unwound it a little bit so you can see all the subunits. And this is very similar to the voltage-gated calcium channel, except it's a voltage-gated sodium channel, VGSC, voltage-gated sodium channel. This does not use a tether, which is the main difference between it and the voltage-gated calcium channel, so no tether. But the nice thing is that they both have a voltage sensor, and it's on the same subunit. It's on subunit 4. So there is a voltage sensor. A voltage sensor. I'm circling all the voltage sensors. Okay. So what's exactly happening here? Well, you could imagine that what's happening is I'm sort of getting all this sodium that built up. Why is it building up? Well, when acetylcholine bound to the receptor, the sodium channel opened and it moves through, right? That's why sodium's accumulating. So I have all this sodium that's starting to accumulate, and hopefully it's making a little bit of sense by now. Well, sodium's going to accumulate, okay, in this picture, right? If this is the membrane, right? You could sort of imagine sodium's accumulating over here, right? Sodium eventually, right, went through the acetylcholine receptor, so sodium's accumulating here. And once again, there's a voltage sensor, okay? So this voltage sensor is going to behave identically to the one we saw in the voltage-gated calcium channel. Again, it's sensitive to all this positive character that's building up around here, right? It's sensitive to all that positive character. And so once it senses all that positivity, what happens? You guessed it. The channel opens, but this time sodium moves through. And that's the reason we call it a voltage-gated sodium channel. It's voltage-gated because it's sensitive to charge, right? Positive charge. And it's sodium channel because sodium moves through it. Just like when we had the voltage-gated calcium channel, it's sensitive to voltage, but it moves a calcium ion through, okay? So maybe you kind of see where this might be going. So let's say I have another voltage-gated sodium channel adjacent to it, right? I'm obviously not doing a very good job for obvious reasons, but, you know, sodium's going to move through here, right? And I think you have the intuition. More sodium is going to accumulate, right? And hopefully you can see where we're going with this. There's probably a voltage sensor right here, and you'd be right if you said there was. And there's probably one here too. And one over here. There's basically 4 subunits here, so there's 4 voltage sensors. And you know, you have this positive charge that's accumulating, right? So what happens? Well, the voltage sensors sense the change in voltage, right? Now you have this massive positive character over here. So what happens? Right, sodium is going to move through. Okay, so what you're essentially seeing is you're seeing that sodium is going to move through into the cytosol because that's its direction of diffusion from the extracellular fluid into the cytosol. That's the direction that it spontaneously moves. And then you're going to have voltage sensors on what we call a voltage-gated sodium channel. And they sense the change in voltage and they open. And then, you know, you have more sodium that moves through here. What's the net effect? Well, along the muscle cell membrane, you start to develop a huge amount of positive charge inside the cell. So that's going to lead us to essentially this, okay? In fact, this right here, this is just sort of a continuation of what I was just talking about. Notice here, this is the same thing except a different picture. This is a voltage-gated sodium channel, okay? Notice here I have a voltage sensor. In fact, on each one I have 4, although they only show 2. Okay, and I think you have the intuition that basically through other voltage-gated sodium channels, right, I'm starting to get lots of sodium that moves through here, right? And what's happening? Well, I'm starting to get a lot of sodium that's accumulating here, right? Lots of sodium and lots of positive character are accumulating in the region of these voltage sensors, right? And so when they sense the change in charge, what happens? The channel opens and you move sodium through into the cytosol of the muscle cell, okay? So like I said, what is the net effect? Well, they've even shown it here. You're starting to build up a lot of positive charge in these regions. Okay, you start to build up quite a bit of positive charge in these regions. Okay, and when you build up this positive charge, the voltage sensors sense this, and that causes the protein to change conformation under conditions in which the channel opens and the ion sodium moves through. Okay, so the net effect, key here, the net effect is you start getting lots of positive charge inside the cell. What is that? Well, whenever you start getting influx of positive charge inside the cell, this is what we call depolarization. Okay? So how would I define depolarization to make a student understand this? The way I've always explained it is if there's any process that you can do where you get migration of positive charge inside the cell, that's a depolarization. Okay? In general, there are two ways we can depolarize. I can either move sodium through or I can move calcium through. In this case, the only one I'm dealing with is sodium. Okay? And so you could imagine what's going to happen. Let's suppose I'm going to draw a hypothetical muscle cell membrane. So this is the muscle cell membrane. Let's draw some voltage-gated sodium channels here. Right? Let's draw some voltage-gated sodium channels, and hopefully this little corny illustration will kind of help you understand what's happening. So once again, extracellular fluid out here, cytosol of the muscle in here. Okay, so maybe from previous voltage-gated sodium channels, or even if we're going back a little bit, the ligand-gated sodium channels, basically we have sodium that's moving inside, right? So sodium builds up inside here, right? Sodium builds up, and then I think you have the intuition there's a voltage sensor right there. It's going to sense the positive character, and what happens? Well, sodium moves through from the extracellular fluid into the cytosol of the muscle cell. Well, what's going to happen from there? Well, I'm going to start building up lots of positive charge here, lots of sodium, right? And then what's going to happen? Well, there's another voltage sensor right here, right? And so then you get this buildup of positive character, and this voltage-gated sodium channel senses it. So what does it do? It opens. It opens and sodium moves through, right? So then what happens? Well, you keep, influxing sodium in this region, right? And you start building up all this sodium and all this positive character, right? And on the next one, there is another voltage sensor, right? And it senses the positive charge. So what does it do? Well, it opens, right? And it moves sodium through, right? And then you start building up more sodium, right? From all the influx of sodium, right? And what's happening? You're building up positive character in here, and what happens? Another voltage sensor senses this, right? And so then what happens? Well, as you guessed, sodium moves through, okay? This situation that you have where you're building up all this positive character inside here, these are called depolarizations, and for reasons that we won't go into now, it's essentially only going to move in this direction as shown. So basically what's going to happen is this propagation of opening of all these voltage-gated sodium channels, it's going to propagate in that direction, okay? And that's about as far as we're going to make it for this week. There's going to be some other topics for you to look at that we'll also have videos for. But if you understand this, where we pick up in the next week, it should make a little bit more sense. So make sure to study this stuff. It's not something that comes easy to everyone. So make sure you watch it, keep up with the homework. See you in class.